So we're going to turn over to our audience questions now. First up, we have a question that is asking: Is there real use of Is there real use of self-replicating mRNA? And I would pose that question to either one of you, or both of you, excuse me.
I think there is use. I think it's a little bit difficult to completely control the safety profile of these new technologies, and so I think that agreeing on what the safety toxin biodistribution package will have to be to demonstrate safety will be key in order to then implement those technologies. We are really intrigued because, for example, our AI-driven platform that dictates or suggests what genes to encode for the HSV can be applied to other delivery systems. So I'm absolutely delighted to see new technologies coming, and I will be really intrigued to use them. But I think that is in particular for the self-replicating RNA, we do need to see a little bit more robustness in that safety profile and the packages. I don't know, Karen, what's your opinion on that?
Yeah, I'm kind of in the same place. I need to see more before we know how viable a product it is, but it's well worth the analysis. Yeah.
Thank you. Karen, perhaps this is a good question for you. Are payload size limitations constricting the ability of LNP to address the market need?
Oh, that's a great question. So I don't know for sure what the full payload limit is, because it depends on the LNP itself and its own size and how much it can package. So I do imagine that there will at some point there are some limits on there. I can't give you a precise number the way I can with a viral vector. We know what size their natural genome is. So there are some, and there are some cases where you might need to use more than one vector to deliver a very large gene, for example, which is true of many other of these delivery systems. So there is a limit there. I don't know quite how large it is, but it is certainly most genes can be encoded there. It becomes more of a question if you want to carry multiple payloads.
Great. Okay. Is the immune response from lentivirus in vivo applications too prohibitive to make a therapy?
Yeah, so I don't think so. Again, it depends on a pre-existing immune response. And so for lentiviruses, for the way they're pseudotyped right now, it shouldn't be too great of an issue, but it is possible that you would need different pseudotypes to avoid that. And then, and because it's expected to be a one and done, the immune response you're going to generate with delivering it in vivo should not inhibit its ability to have gotten in and to express. It just meant it means it's not redosable. And so for that, as you do with other vectors like AAV, if you do a clinical trial, your initial dose is intended to be a therapeutic dose. You don't dose below the therapeutic dose and then try and raise it up, because you can't redose the same patient. So you want to give them all doses that are intended to be therapeutic.
Sure. Okay. One more question. Are there additional safety concerns for lentiviral versus AAV? Perhaps, Karen, you could answer that. And then I pose the same question to Francesca. Are there additional safety concerns for HSV versus AAV? But Karen, if you could kick us off.
Yeah, so they have different safety concerns, as it turns out, because of how they're administered and because of their endogenous properties. So for AAV, certainly some of the safety concerns have been for patients in which very high doses have been administered, especially systemically. There's always the question of liver toxicity, which is being addressed, with immunosuppressive agents and with other treatments to try and avoid that. That's one example of where safety is an issue for AAV that we haven't just gotten to today. For lentiviral vectors, for using them ex vivo, biggest safety hurdles that I see are not in the lentivirus itself, but how the patient is treated in order to allow for ex vivo treated for their own stem cells to be reinfused. And to do that, you essentially make room in their bone marrow by giving them by prepping them with immunosuppressive agents and essentially giving them chemotherapy, in some cases high doses. So for those patients, what they're treated with to prepare them to accept the ex vivo cells to come back in and to repopulate, that is a very intense procedure. And there's effort being done to make it less intense. But it's essentially like doing a full bone marrow transplant, is that pre-treatment. So that's an area where there needs to be a lot of work in being able to reduce the effects of that and being able to potentially reduce the doses.
Sure. And Francesca, same question to you for HSV.
So both for the adenovirus and for the HSV, we don't have these hurdles. I mean, in terms of safety and biodistribution is extremely localized and contained. So for the adenoviruses, the classical, tox profile that we see in patient is a little bit of flu-like syndrome that is really the activation is just a reflection of the activation of the immune system that is systemic. Even if you deliver locally, you do have these T cells that start proliferating and they tend to go around the body. So we have a raise in the cytokines, but it's never a big like cytokine release syndrome, right? So we're talking about a little bit of fever. Some patient can have some chills, and we don't observe it in all the patients either. So it's extremely self-limiting also. It lasts like 48 hours, no more than that. For the HSV, we've seen, for example, in the Replimune case, so virus is delivered like T-VEC on the skin. Mainly they have like a melanoma or squamous cell carcinoma. We see inflammation in the area, and again, very little systemic level of activation. For CAN3110, that is the virus that I described before, the replication competent vector, the FDA was very concerned at the beginning because we were talking about delivering in the brain of this patient a replication competent HSV. So everybody was worried that we're going to have encephalitis. And despite the fact that we had a very clear preclinical tox, so we did the GMP tox to go to the IND, the FDA asked us to do a very slow dose escalation phase 1 study. We've done it. We started at 1 × 10⁶, and we painstakingly escalated half log at a time. And we've seen that the patient tolerated extremely well to up 1 × 10⁹. Now we set it to 1 × 10⁸, just because the 1 × 10⁹ had some inflammation that was like that obviously in the brain, that is a constrained organ because of the bones. It causes more problems because you have swelling and so on. You don't want that. So 1 × 10⁸ was perfectly tolerated, and this is where we've done our expansion cohort for the patient, and we've seen then clinical responses too. But yeah, I think that in general, both adenoviruses and HSV have got a very good profile of safety and tolerability. In fact, they're very good at being added to standard of care because they don't add the significant toxicity to standard of care.